everyone, and welcome to the podcast. Welcome to a very exciting episode 109. Today's show is the first in a new series on a new clade of the protostome animals. In the previous series, we explored the amazing physiology and evolutionary diversity of the mollusks, those most basal of protostomes. And now we're going to look at the next branch on the protostome family tree, except this particular branch on the tree of life is absurdly big. It's easily the biggest branch on the eukaryotic tree of life, containing a majority, by far, of all eukaryotic species. The mollusk branch on the family tree is large, make no doubt about it. There's thousands of species of gastropod and bivalve, and hundreds of species of cephalopod. All told, the mollusk phylum contains about 85,000 living species. For comparison, the reptiles, the birds, and the mammals each contain about 10 to 11,000 species. So the mollusk phylum it's pretty big, but the mollusk branch is easily overshadowed by the arthropod branch of the tree of life. The arthropods are, without question, the most diverse clade of eukaryotic animals on the planet. For example, consider the weevil of the family Circulinidae. This is a family, not a phylum, so taxonomically speaking, it's much more constrained and specific than the mollusks, and yet there are approximately 83,000 species of weevil, which is about the same as the entire number of living mollusk species. Now consider this. The 83,000 species of weevil in the Circulinidae family are grouped in the Coleoptera order. You might recognize the Coleoptera as the beetles. This is the largest order of animals on the planet, with almost 500,000 species that are both currently living and known to science. That represents about 25% of all known extant animal species. In other words, if you were going to randomly select one of the more than 1 million known animal species on the planet, there's a 1 in 4 chance that your random selection is going to be a beetle, something from Coleoptera. That's crazy, but it doesn't end there. The Coleoptera order is part of the Insecta class, which itself has something like 1 million known species and an estimated 6 to 10 million species total out there in the wild, and it represents about 90% of all animal life. 9 out of 10 animals are insects. And that's just the insects, or if you want to expand out a little bit, the hexapods, and these are just one of the 4 classes of arthropods. This should give you a sense of scale of just how numerous and abundant and diverse the arthropods really are compared to pretty much everything else. Now we're going to discuss the hexapods, and the insects specifically, sometime in the future. But before we do, we have to explore the more basal arthropod groups, including the crustaceans, the myriapods, and the arachnids. There's a colossal amount of diversity within the phylum Arthropoda, and I would never be able to sufficiently cover all of these animals in a single series, like I did with the phylum Mollusca. So instead, I'll be covering the phylum Arthropoda in 4 separate series, one covering each major clade from the most basal to the most derived. This new series that we're beginning today will be all about the most basal arthropods, the arachnids and their relatives. This first episode will be all about the emergence and evolutionary history of the arachnids, including a look at their most basal lineages that still creep and crawl along the surface of the earth today. As the arachnids are the most basal arthropod clade, any study of their early evolution will necessarily be a study of the deep evolutionary history of all the arthropods, of the clade as a whole, going all the way back to the Cambrian, or even earlier, when the ancestors of arthropods diverged from the ancestors of what would become their mollusk cousins. Now, as I explained in my episode on mollusk evolution, the fossil record going back this far is frustratingly sparse. Soft tissues rarely fossilize, and even when they do, fossils from over 500 million years ago have ample time to be destroyed by erosion, or the trampling feet of larger animals, or the growth of roots, or rainfall, or gradual erosion from wind, or any other kind of natural process that can gradually wear away and destroy fossils. With respect to the arthropods specifically, their exoskeletons are certainly harder than the soft tissues of a slug or an octopus, but they're not nearly as hard as the mineralized shell of a snail or a nautilus. This means that the arthropods, especially the earlier arthropods that had softer exoskeletons, were just not very likely to fossilize in the first place, and this relative lack of fossils is consequential. Our understanding of the early relationships between all of these ancestral groups is hazy at best. With the advent of genetic and molecular analysis methods, older phylogenies, built on morphological research developed from countless painstaking hours behind the microscope, have turned out to be almost completely useless. And even worse, in many cases, genetic and molecular analysis hasn't provided clear answers. The phylogeny of crustaceans, for example, was already a confusing mess before genetic analysis. After genetic analysis, one could argue that it actually makes less sense, and we're less confident about how to arrange the crustacean phylogeny than we were before. Furthermore, as more research is conducted and new papers are published, the phylogenies are tweaked and adjusted and changed in accordance with the latest data. For example, consider the group Lobopodia. We'll talk about the lobopods more in a few minutes, but for now, all you need to understand is that these are the ancestral pan-arthropods, and over the years, there's been much debate about whether the lobopods are all extinct, or if they should include living non-arthropod groups like the velvet worms and the tardigrades. When scientists use different definitions of these phylogenetic clades, it complicates any attempt at an organized, objective, singular, universally agreed upon phylogeny. This is a really important clarification that you should be aware of, because when I did the research for this episode, I was trying to use modern sources. I avoided phylogenetic molecular research papers that were more than 15 years old, and I tried to seek out and prioritize papers that were published within the last 3 to 5 years. Unfortunately, the field moves so fast, and new discoveries can be so disruptive that some of the papers at the older edge of that 3 to 5 year range were having their results overturned or put into a different context by new discoveries that were described in papers published at the more recent end of that 3 to 5 year range. As I was writing the script, things were changing and being updated, and I had to go back and fix certain details. It was kinda surreal, but that goes to show you just how fast this field moves. So, when it comes to phylogenies and paleontological theories on ancestry and relatedness among these protostomes, what I present to you here in this episode is my best attempt to describe the current state of knowledge. But please understand that a new fossil discovery or a new genetic analysis could change some details here or there, even some big picture things. There could very well be a paper that comes out like a month after I post this episode that has some revolutionary finding about spider evolution or spider anatomy or something like that. And so when we're talking about fields of research where the study material is scarce like this, like early arthropod fossils, new discoveries can be really disruptive, and they can cause major shifts or changes in our understanding. To put this another way, imagine a spoonful of water, and you put a single drop of food coloring in that spoonful of water. Well, that single drop of food coloring is going to cause a major change. It's a small body of water, and that drop of food coloring will deeply stain the whole thing. Compare that to a field that's, let's say, more developed, and there's a wider range and breadth and depth of research, in different subfields and subdisciplines and aspects of whatever phenomenon they're looking at. This is like a river or the ocean. If you were to put a drop of food coloring in a river or the ocean, It's not going to change that much. That new discovery might be important, but so much is already known that new discoveries aren't really going to disrupt the status quo too much. With all that being said, with all that housekeeping out of the way, let's begin our story of arachnid evolution. The beginning of this story takes place at the onset of the Cambrian explosion, around 542 million years ago. At this point in time, life on Earth has been evolving for approximately 3 billion years, but for most of this time, life was unicellular, single-celled. But in the later eons of these 3 billion years, the complexity of life, particularly eukaryotic life, began to accelerate. Evolutionary processes worked upon numerous ancestral traits, like scientists building off of the work of their predecessors over the centuries. Endosymbiosis of the mitochondrion provided enough energy to enable specialization and multicellularity, which led to the first true eukaryotes 1 to 2 billion years ago. The eukaryotes, with their greater complexity and energy efficiency, grew in number and eventually became the primary movers of nutrients across time and space, across habitats and landscapes. In effect, they achieved ecological dominance over the prokaryotes sometime around 800 million years ago. Within this emergent, newly ecologically dominating eukaryotic lineage, a major evolutionary split would occur about 610 million years ago. This divergence would create 2 major lineages, the deuterostomes and the protostomes. The deuterostomes are generally larger, more complex animals, including the starfish and sea cucumbers, as well as all of the chordates, which includes all of the vertebrates like fish and mammals and everything else with a spinal column. We'll get to all of these groups in future series, in due time, but for now, the protostomes would go on another evolutionary path. They would diverge wildly, producing the overwhelming majority of the world's animal species. We already explored the clade mollusca and their Spiralia allies, which are themselves the most basal protostomes. In summary, the earliest mollusks descended from the Lophotrochozoans, tube-like or slug-like creatures that had a batch of tentacles around their mouth and a shell forming on their back. Early arthropods had a similarly slug-like or tube-like body shape and a marine habitat. They lived in the ocean. But, more importantly, instead of developing a mineralized shell like a shield on their back, the ancestral arthropods evolved body segmentation, a trait that mollusks never developed. Segmentation, and for that matter, body parts with joints, like arms and legs, probably first evolved on the slug or tube-like form as a means to maintain the ability to move while still evolving a hardening exoskeleton. This jointed, armored body form would preserve the animal's ability to flex and bend its body, kind of like how a medieval European suit of armor has lots of overlapping plates at the joints to enable as large a range of movement as possible. Furthermore, each body segment had an identical pair of limbs and various repeating organs, typically built with a consistent toolbox of genes that are reused and slightly tailored for every segment, so the entire body seems repetitive or modular to some degree. The myriapods take this simple form to an extreme, evolving numerous repeating segments, sometimes hundreds. In all the arthropods, some of these body segments, and the limbs, tissues, and organs within them, would experience mutations and divergences. The limb might be repurposed or modified to serve some new function. Think something along the lines of a leg near the head or the front of the animal being transformed into a mandible or a pedipalp to help with feeding, or even a claw or an antenna of some kind. If the mutated appendage doesn't work, that lineage may die out. But there's countless more, their redundant limbs experimenting with repurposed or altered functions. Through this mechanism of repetition and redundancy, especially through the gene toolkit and gene expression systems, segmentation provided ample room for evolution to play around. Segmentation became a way to rapidly develop adaptations and subsequent biodiversity, which would have major implications for the future evolution of the arthropods. Around 530 million years ago, so just a few million years after the Cambrian explosion, a new lineage emerged from these early arthropod ancestors called the Ecdysozoa. This lineage is defined by the ability of its member species to molt, to regularly shed their exoskeleton. This is a very fascinating process, and it demonstrates some of the initial differences in the evolutionary trajectory between the ancestral arthropods and the ancestral mollusks. In the mollusks with spiral shells, for example, like the gastropods and the nautilids, the shell begins growing from an initial chamber, and then it spirals outwards, getting larger and larger as the opening of the shell grows, spiraling away from the initial starting point. As the mollusk grows larger, the shell can be grown with it, with mineralized tissue being deposited along the open end of the spiraling tube. Thus, the shell isn't necessarily a constraint on the growth of the mollusk. It's open-ended, literally and figuratively here. The shell can grow continuously along with the mollusk, and we've seen some fossils that are quite enormous. Now, these shells still require a ton of nutrients, like minerals, so there may be a constraint on growth in that sense, in the ability for the mollusk to get enough food to sustain the shell's growth and maintenance. Maybe the shell's demand for nutrients slows the growth of softer tissues, but from an overall functional morphological perspective, the spiral shell design itself doesn't constrain the ultimate growth of the soft tissues. This is a really important detail, because the early arthropods did not have coiling shells capable of supporting indefinite growth of the soft tissue. Instead of a coiling shell that the soft tissue kind of poked out of, the early arthropods evolved firmer external tissue. It isn't mineralized, but it does form a stiff exoskeleton. And while this offers some mechanical protection and a structure upon which muscles can exert a force to move the body, the exoskeleton itself is not very flexible. It's not stretchy. And this is a problem, because if the soft internal tissues of the arthropod keep growing, they'll push up against the harder, inflexible exoskeleton. Growth of the soft tissues is thus constrained. The Ecdysozoa solved this problem when they evolved the ability to molt. At specific points in their adolescence, and in some groups at regular points throughout maturity, the current exoskeleton will break apart along a series of built-in seams. This rupture of the older, worn-out, tight-fitting exoskeleton allows the arthropod to burst out of it with a shiny, often slimy, newly grown exoskeletal layer underneath. This newly grown exoskeleton may be somewhat flexible and will take a short time to harden completely. As this happens, while the new exoskeleton is still flexible, the soft internal tissues will plump up with water, and the arthropod will expand a little bit. It gets a little bit bigger before the new exoskeleton hardens into place. A typical arthropod will go through multiple molting events in their life, and depending on the species and the number of moltings, the older adults can be huge, monstrous even. This is particularly true for crustaceans, like lobsters and certain kinds of crabs. Some of the more basal Ecdysozoa include the Priapulida, the so-called penis worms, named after their peachy skin color and their phallic shape. There's also the Caenorhyncha, which are called mud dragons, despite their very diminutive size, and all the various microscopic nematodes and nematomorpha that live everywhere and cover everything. Another Ecdysozoa clade that emerged around the beginning of the Cambrian would become known as the Panarthropoda. Key among the Panarthropoda are the Lobopodia. In essence, the Panarthropoda is a clade that includes all the arthropods and all of their closest cousins, including the onychophoran velvet worms and the tardigrades. You should remember these panarthropod cousins, as we'll get to them in just a minute. The Lobopoda represent what is arguably the most basal or the most ancestral panarthropod. These are the quintessential primordial arthropods with segmented bodies and repeating pairs of limbs. Their fossils show the development of hard tissues like claws, fangs, and armor plates, all protruding from or integrated into the classic exoskeleton. These lobopodians emerged in the mid-Cambrian, and although they would die out several 100 million years later, many of their diverging descendants would live on into the modern day. Some of the most famous mid-Cambrian lobopods include the Aischiaia pedunculata, a fat worm-like creature with 10 body segments, 10 pairs of limbs, and around its mouth a half-dozen finger-like protrusions that are reminiscent of their Lophotrochozoan cousins. It's believed that Aischiaia fed on sponges, which, due to their immobility, their soft, unarmored tissue, and total lack of defenses, would have been an optimal and easy food source in the mid-Cambrian. Aischiaia had a body form remarkably similar to the modern onychophorans, the velvet worms. By studying the velvet worms, we can make inferences about the behavior and ecology of Aischiaia. Briefly, these are predatory insectivorous invertebrates that live in tropical regions, primarily in the Amazon and in Mesoamerica, but certain specimens, a few species, have also been found in geographically limited or isolated regions of Southeast Asia. The velvet worms do not have an exoskeleton. They lost that trait, and now they're soft-bodied, which puts them at greater risk of mechanical damage. But due to their gray or subdued color and their slow, ponderous, almost unnoticeable movement style, they easily melt into the shadows and the underbrush of their habitats, and they just go unnoticed by predators like centipedes, spiders, birds, and small reptiles and mammals. Most fascinatingly, the velvet worms can eject a sticky slime onto their insect prey, which immobilizes them. This is really helpful, as the velvet worms have very poor vision, very poor hearing, and they don't even seem to track their prey by scent, which is really unusual. Instead, they use the very subtle changes in air pressure from the movement of their prey to track them. When they get close to their prey, they very gently touch the prey animal with their fuzzy antenna to measure it and deduce if it's worth it to make an attack. This is where the immobilizing spray juice comes in, allowing the slow, ponderous velvet worm to take its time, meandering over to its prey, and eat it at a leisurely pace. Unfortunately, due to their soft tissues, they don't preserve well at all, and the fossil record beyond 40 million years is largely absent of unambiguous onychophoran fossils. This isn't to say that they didn't exist more than 40 million years ago, just that they're really unlikely to fossilize in the first place, so any potential fossils are going to be really rare, and we haven't found any that are older than that. A similar issue exists for the tardigrades, or the water bears. These are very small, 6-legged arthropods, believed to have evolved from much larger ancestors at some point in the distant past. It's believed that the tardigrade lineage branched off hundreds of millions of years ago as part of one of those later generations of lobopodian divergences. The tardigrades are widespread. They're important pioneer species that move into unsettled biomes and participate in gradually making the chemical environment more suitable for life, while also attracting other life forms like parasites and predators. And despite the tardigrades having existed for so long, their relatively soft bodies and very small size means fossils are also extremely rare and hard to come by. The oldest tardigrade fossil we have is a specimen of the Milnesium genus that's barely 90 million years old, and just like the velvet worms, we have strong genetic evidence that suggests that they've been around a lot longer than that. Molecular clock research, for example, suggests that some of these lineages should be hundreds of millions of years old, but the fossil evidence that we have is so limited that the oldest we can trace them back to is not even 100 million years old. Okay, so we saw the evolution of the ecdysozoa and the emergence and diversification of the lobopods. As this was going on, as these lobopods diversified, another important lineage emerged. It's either a basal lobopod or an ecdysozoan sister group. It's not quite resolved, but in any case, it represents the immediate ancestors of the arthropods. the first lineage of true arthropods to emerge from these ancestors were the Chelicerata, which were distinguished by the evolution of the chelicera, a small pair of appendages mounted on the front of the head, which are used to handle food and bring it to the mouth. Another pair of similar appendages were also evolved alongside the chelicera. This second pair of limbs are the pedipalps, which have evolved different shapes for different purposes in different species. Some chelicerates have pedipalps that contain sensory organs, or sex organs, while in others they've been adapted into pincers, from small, thorny, finger-like structures to enormous pincer claws. Both the chelicera and the pedipalps are modified limb structures protruding away from the face and crowding around the mouth. Anyway, one of the first subgroups to emerge from this chelicerate lineage were the Pycnogonida, or the sea spiders. Now before we get into these guys, just be aware that within that statement, I've stumbled into a long, ongoing controversy. The phylogenetic placement of the sea spiders of these Pycnogonida has apparently been in dispute for decades. The sea spiders are traditionally considered to be part of the Chelicerata. Specifically, they represent the most basal lineage of the Chelicerata that still has living members. But there's genetic evidence against this idea, suggesting instead that Pycnogonida emerged shortly before the Chelicerata, meaning the sea spiders are actually not true arthropods, and they may be, in fact, a sister lineage to all of the arthropods. In any case, the fossil record is scant, and not much is known about the early pycnogonids, but they're suspected of having first emerged in the late Cambrian, around 500 to 490 million years ago. Much like the onychophorans, we can make inferences about the ancestors by looking at their modern descendants. Today, the world's oceans contain over 1,300 species of sea spider, and nearly all of them are characterized by a relatively small body and disproportionately long legs. Most have 8 legs, but a small number of species have 10 or even 12 legs. They can have 1 or 2 pairs of eyes, but species that live in the deep sea, where it's pitch dark all the time, may have no eyes. The long legs serve multiple purposes. In addition to your basic locomotion, such as swimming over the seafloor and scuttling across rocks and coral reefs, the large surface area of the legs also serves an important purpose in gas exchange. Instead of a respiratory system, gases are absorbed through the legs, where they quickly go into the organs. The body of the sea spider is usually so small that organs and other internal parts are squished and pushed into the legs, making room alongside long, thin muscle fibers. What's even stranger is that these newly hatched sea spider larvae will immediately attach to the father's legs, and he'll carry them around until they grow large enough and they grow enough legs to detach from him and walk around on their own. In their habitats, the sea spiders are carnivores, either active predators, parasites, or scavengers, who roam around using their proboscis to puncture the skin of their soft-bodied prey and suck out their juicy, meaty innards. This soft-bodied prey includes sponges, various worms, bryozoans, and corals. They're spindly aquatic spiders that clean up the fleshy debris and prey on soft-bodied, usually defenseless, filter-feeding animals. They do have several predators, including certain fish, crabs, starfish, and even some species of shorebirds. Okay, let's step back for a minute and recap where we are, evolutionarily. 610 million years ago, the deuterostomes and protostomes split. Following the protostome lineage for a while, we see the development of exoskeletons, segmentation, and then molting, with the emergence of the ecdysozoa 530 million years ago. Among the lineages that emerged here, among these molting protostomes, were the sea spiders around 500 to 490 million years ago, and shortly after them came the Xiphosaura, or the horseshoe crabs, about 445 million years ago. Now, many other new related lineages would emerge and then die out in this time period, their fossils depicting a morphological menagerie of features on a common body system. There's a downward-facing mouth, round armor plating over the body, protecting the legs underneath, and a long tail. Different genera display different shapes of the shell, where they possess spikes either on top or along the front edge, or the midsection of the body is extended laterally, or there's some other slight derivation like that. Of all these forms, of all these different species of horseshoe crabs emerging and proliferating and diversifying, today, only the Limulidae exists. That's the only family that still remains, and it contains just 4 species of horseshoe crabs. The Limulidae family emerged from their Xiphosauran ancestors about 330 million years ago, or sometime in the mid-Cretaceous period. That's more than 100 million years of horseshoe crab evolution, where all of those diversifying lineages that were experimented with have all died out, and only the single Limulidae family remains. Curiously, they appear almost identical to their earliest ancestors, which are hundreds of millions of years old. It's a testament to how robust and effective their body form is for their habitat, where they can crawl along the seafloor in relative safety, their armor plating creating a safe cavity around the mouth which covers and traps prey, which is then consumed and ripped up in the gizzard. They typically feed on worms, on clams, and small crabs and prawns. Now, aside from the 445 million year old Xiphosauran branch, the rest of the chelicerates continued to evolve and diversify throughout the Silurian period. Many of these species that emerged here were just more permutations of horseshoe crabs, and they too have all gone extinct. But one lineage, which emerged around 10 million years after the Xiphosaurans, so around 435 million years ago, this lineage would break out from this evolutionary pattern and colonize new habitats, new ecological niches. It would produce new body forms and find new prey animals to eat. This lineage is Arachnida, the true arachnids. By the Devonian period, around 420 to 400 million years ago, many diversifying lineages of early arachnid had left the sea and colonized dry land. They were participating in the ongoing surge in complexity of terrestrial ecosystems. For the arachnids, this transition onto dry land literally opened up a whole new world of resources and habitats, but it also brought with it new predators, but also new types of prey. The diversification that this enabled was explosive, but the fossil record is sparse, and genetic analyses are frustratingly inconclusive. Without going too much into the weeds, the early history of arachnid evolution is not clear. Lots of different groups emerged, but the precise order and arrangement of these groups has not been conclusively demonstrated. We'll be going over some of the major groups of arachnids throughout the rest of the series, but before we wrap up today's episode, I wanted to look at 3 arachnid groups that are generally considered early primitive representatives of their clade. The Opiliones, also known as daddy longlegs, the reddish-brown Rhicinulae spiders, and the Solifugae, also known as the camel spiders or the sun spiders. Take note that none of these are actually true spiders. They're all older, more basal cousins of Araneae, the true spiders in the class Arachnida. With that being said, let's take a look at these 3 primitive arachnid groups to really set the stage for the rest of the arachnid class, for all of the orders that we're going to cover in the rest of the series. So let's start with the harvestmen, the daddy longlegs, the Opiliones. This clade emerged around 410 million years ago and transitioned from sea to land. Ancient fossils in very well-preserved sediments show tracheal and reproductive structures that are nearly identical to modern-day harvestmen. I've always thought that these daddy longlegs kind of looked like sea spiders that dried out and shriveled up on land, considering they also have small bodies and disproportionately long legs. And it looks like, despite having diversified to produce a modern lineage with over 6,650 species, it looks as if they've maintained that general physiology for pretty much all of their existence. However, the harvestmen, these opiliones, they differ from many or all other arachnids in several fascinating ways. Most arachnids have venomous chelicera, but the harvestmen do not. Many arachnids have silk glands and can spin webs, but the harvestmen don't have those glands, and they can't spin webs. Most arachnids have 3 to 4 pairs of eyes, but the harvestmen only have 1 pair of eyes. A very small number of species have 2 pairs of eyes, or no eyes at all. The opiliones have evolved to use their second pair of legs kind of like antennae to sense the environment, and as a result, this second pair of legs has been evolving to be even longer and narrower than the rest of their already long and narrow legs. They breathe through trachea instead of book lungs. They use their uniquely pincer-like chelicera to rip up their food and ingest particles of meat or plant tissue, instead of just slurping up liquids. And on that note, the clade demonstrates a very broad range in their diet, from the usual arachnid fare of small insects, but also plants, fungi, dead animals, feces, and other detritus. On the topic of opiliones reproduction, the males are particularly unique among arachnids. Not only do they have a penis and practice direct copulation and internal fertilization, which is unusual among the arachnids, they engage in post-copulatory cuddle-like behaviors, and then exclusively guard the eggs after they're laid. This is very uncommon among arachnids, especially among males. Perhaps the most visually characteristic feature of the clade, aside from their legs, is the broad connection between the head, or the cephalothorax, and the abdomen. This connection is not a narrow joint. It's broad. There's a wide border between the head and abdomen, so their bodies form a cohesive, circular, or oval shape. All of these features of the harvestmen are unique to them, or at least they're very rare among the rest of the arachnids. It's also worth noting the diversity of ecological strategies that they use. They include both nocturnal and diurnal species. They include carnivores, herbivores, omnivores, and detrivores. Some of the harvestmen are solitary, but most of them are highly social, and they're quick to group up for the collective benefits, like stronger pheromone signals, improved chances of finding food, and when it comes to avoiding predators, there's safety in numbers. The second group of primitive arachnids, and closely related to the opiliones according to most genetic analyses, are the Rhicinulae. This small group of arachnids contains a mere 76 living species, so just a little more than 10% of what we saw in the opiliones. And while the Rhicinulae have much more in common with their more derived arachnid cousins, like being entirely carnivorous predators with much greater morphological resemblance to the true spiders of Araneae, the Rhicinulae also retain some more basal traits, like those seen in the opiliones, like chelicera equipped with pincers, breathing through a trachea, a preference for large groups or collectives, and the second pair of legs are specialized for use as antennae. Unlike the opiliones, the body and legs of the Rhicinulae are thick and plump. Their waist, the connection between head and abdomen, is not broad, but is actually quite narrow. The abdomen itself is elongated into a bulbous oval shape, often demarcated with lateral plates or ridges running along the back. Ancestral Rhicinulae used to have eyes, but all 76 living species are completely eyeless, although there's a small number of species that retain clumps of light-sensitive tissue where the ancestral eyes used to be. Instead of eyes, a thin armor plate or hood sits over the head and can be retracted down to cover the face and the mouth. Aside from the chelicera, they also possess a larger pair of forward limbs, pedipalps, that also have small pincers and sensory structures. Now, while their anatomy has been extensively documented, not a whole lot is actually known about their behavior or life strategy. What limited research there is on the Rhicinulae ecology suggests that they're largely nocturnal predators that hunt in dark terrestrial habitats, like the floor of a rainforest or a cave. Here, they seek out and feed on smaller invertebrates, like little insects and worms. Do you remember earlier that I said that this early arachnid taxonomy was not clear? When it comes to organizing species and creating a phylogeny of who emerged when and in relation to who, we don't really have that figured out yet. Associations based on morphology, life stages, or behavior are often disproven by later genomic research. For example, the Rhicinulae have a larval stage where the individual has 6 legs and develops the 7th and 8th legs during molting and maturation. This is a trait that's shared with the Acari, a group that includes the mites and the ticks. Now at first, this was thought to be evidence of close relatedness between the Rhicinulae and the mites and ticks, until, of course, subsequent genetic analysis suggested otherwise. A more recent genetic analysis has placed Rhicinulae as members of Xiphosaura alongside the horseshoe crabs, while another recent study has found them to be a closely related sister lineage to the Opilionies. Like I said, we know that these are primitive arachnid groups, but their precise arrangement remains a mystery, and genetic analyses often provide conflicting, confusing results. The third group of basal arachnids are the Solifugae, often called camel spiders or sun spiders, despite being neither a true spider or a true scorpion. The Solifugae, or Solifugae, emerged in the late Carboniferous, around the same time as the Araneae, the true spiders, so I suppose it's not quite accurate to call the Solifugae primitive or basal. I'm including them here more so because their arrangement in the arachnid phylogeny is uncertain, and most genetic analyses seem to place them close to or in between the Opilionies and the Rhicinulae. They also share many physiological traits with one or both of these groups. For example, the Solifugae camel spiders breathe through a trachea instead of book lungs. They do not have silk glands, and they don't spin webs. Their chelicera aren't venomous. They have elongated abdomens, just like the Rhicinulae, but even more so, they typically have a single pair of eyes, mounted in the center of their head, facing out to the sides. But many species have no eyes at all, like the Opilionies. However, a small number of Solifugae spiders have a more sophisticated eye structure that appears to be in the process of evolving through a transition from a compound eye to a simple eye. This seems to imply they're being naturally selected for visual clarity and image formation, which are very useful tools for hunting, especially when you live in a habitat with lots of sunlight. The Solifugae camel spiders can be found mostly in arid deserts and scrubland biomes. Although some species are adapted to slightly wetter grasslands and even forests, they universally seem to prefer warmer temperatures and lower humidity. The Solifugae have 2 characteristic features. They've evolved absolutely massive pedipalps that resemble a pair of arms or a 5th pair of legs coming out of their face. These are used like antennae for sensory purposes, as well as for day-to-day activities like capturing prey, food handling, and fighting off other camel spiders. They've also evolved large, overdeveloped chelicera with surprisingly powerful pincers. The muscles are very thick, and the actual appendages are rather big and long, so that the chelicera gives the head a prolonged, almost triangular shape. The chelicera are also lined with variable numbers of teeth, or spikes, with an angled cutting edge that resembles scissors. These brutal chelicera are perfect for the camel spider's lifestyle and hunting strategy. As the desert biomes tend to have a low density of animal life, the Solifugae must opportunistically feed whenever they come across a smaller animal, like a beetle, a termite, or a smaller arachnid. This opportunistic hunting style sometimes leads to risky, but often successful attacks on larger prey, like rodents, lizards, and other small vertebrates. Once a prey animal has been identified and captured using these strong leg-like pedipalps, the unstoppably powerful chelicera are then used to physically dismember and destroy the prey animal, until it's sufficiently pulverized and sliced up that the camel spider can suck up its juices. Alright, so I think this is a good stopping point for today's episode, and the next episode will continue this exploration of the murky, mysterious beginnings of arachnid diversification by covering 2 more basal lineages. Much like the Opiliones, the Ricynulae, and the Solifugae, these 2 lineages that we're going to cover in the next episode also move around a lot on the family tree depending on which genetic analysis you're referencing, with some finding close relatedness between them, while others find a greater distance between them. These 2 lineages are the Acariforms and the Parasitiforms, which I thought deserved their own episode due to their widespread specialization into a parasitic ecological niche. Some of them are scavengers, or straight-up carnivores or omnivores, but all of them are fascinating in their own way. If this sounds interesting to you, if it sounds like something you want to take a deep dive into, then hit that subscribe button and hit the bell for notifications when I post something new. If you like this episode, then hit the like button, or if you're listening through the podcast app, then give the show a 5-star rating. It really helps with the algorithms, and of course that means that more people can find the show and enjoy their own exciting dive into the world of the biologic. And as always, thank you for listening.